wonderful Friday. The rain is not coming here to Southern California, but what we have is really a group of extremely talented vision research here in Southern California, and one of them will be today from San Diego visiting us. So really, it is a very special place with so many people interested in eye and processes in the eye. So without any further delay, again, just remember to share with your family, with your friends, your enemies, the link for this meeting. Whoever you just desire, just do that. And now we're going to Susie.
Good morning, everyone. It is a great pleasure for me to introduce today's speaker, Dr. Radha Ayyagari. Dr. Ayyagari is a professor in the Department of Ophthalmology and Pathology and Chief of Ophthalmic Molecular Diagnostic Laboratory at San Diego. Dr. Ayyagari completed her undergraduate study at Andhra University, her graduate studies at Kenya University in India, and then she completed a fellowship in ophthalmic molecular genetics at the National Eye Institute in Bethesda. In 2004, she became an assistant professor in the Department of Ophthalmology and Visual Sciences at the University of Michigan, Ann Arbor. And since 2007, she has been a professor at UC San Diego. Dr. Ayyagari and her lab have made significant contributions to understanding of molecular genetics of macular and retinal dystrophy, as well as biological mechanisms underlying retinal disease. Dr. Ayyagari also received many awards, including Sybil B. Barrington Scholar Award, University of Michigan Research Faculty Recognition Award, and the Lou Wasserman Award. With this, please welcome Dr. Ayyagari.
Thank you, Susie, for your kind introduction. So that's very generous of you. I like to thank Grazina, Dorota, and Chris for inviting me to participate in this seminar series and share some of the work we have been carrying out. So now I'm going to share my screen, I guess, right? Chris? Okay.
Okay.
Do you see presentation mode? No, right? Okay.
Now it's perfect.
That's good?
Perfect.
All right. So at least I could share my screen, the first step. So today I'll be talking about inherited retinal dystrophies. This is something which I have been working on. So when I'm talking about inherited retinal degeneration, so this includes, I'm going to just give me a minute. I'm going to move the people's faces from my screen so that way I won't cover the, okay. Okay, all right. So when I'm talking about inherited retinal degeneration, so these include macular degenerations that result in central vision loss and retinal degeneration, which is a mainly peripheral retinal degeneration. So these include LCA, other recessive or dominant retinal degenerative conditions. So collectively, I'll be referring them as inherited retinal degeneration. So as you know, like there are several issues that complicate the diagnosis of these diseases, both clinically and also with reference to molecular diagnosis. One is, some of these I listed here. So different modes of inheritance, phenotypic heterogeneity, genetic heterogeneity, variable penetrance, variable expression, and overlap in their clinical phenotype. So this is a slide most of you are familiar with. So at this point, like there are at least 307 independent loci that are associated with retinal degenerative diseases. And among them, actually, we know about 280 of those genes. And mutations in these genes, they contribute to about 60% of cases. So today, in my talk, what I'll be covering is mainly our interest is understanding the genetic landscape of these diseases. And one of the primary goals in this is to identify missing causality. So we call it elusive genes project. Because all of us have been studying these retinal degenerative diseases and then ended up now with after all these searches, so we ended up with pedigrees where it is challenging to find mutations, and then that's what is our focus. And the second half of the talk, I'll be talking about understanding the disease mechanism, for which I'll be using the late onset retinal degeneration as a model. Okay. So we have sequenced about 454 whole genomes. So this was done by HLI, which is a former Craig Venter organization. And so these individuals are from 120 pedigrees. And these pedigrees are from 3 different populations. One is a Mexican population from the Yucatan Peninsula, and Punjab Sindh region, pedigrees from Pakistan, from the Punjab Sindh region of Pakistan. So these 2 populations are understudied populations. And the other set of pedigrees are from the Caucasian population, which are recruited from the US. So we utilized Illumina methodologies using Illumina X10, and then we sequenced these at 30x depth. So to assemble this sequence and analyze it, so we put together a pipeline using many of the tools that are publicly available. We also put together a pipeline to interpret the variants or identify variants in these sequences, to identify large deletions, small deletions, and then single nucleotide variants, variants in the regulatory regions, and predicting the impact of these variants. So these pipelines we have already published, and we will be happy to share with anybody if anyone is interested in. I'm not going to go into the details today in the interest of time. In each individual, we observed about 4 to 4.5 million variants. So now the challenge is we need to identify the single variant or 2 variants in case of compound heterozygous mutations that are the underlying cause of disease. So for this, we established a filtering strategy. And here I'm going to talk about a pedigree. Here, this is an affected individual, and this is her unaffected sibling and father and mother. So this individual has inherited retinal dystrophy, and there is no known family history of disease in this individual. So these are the clinical details, which I'm not going to go into these details at this point. But she developed the age of onset is 13 years in this individual. We sequenced the whole genomes of the patient and her unaffected sibling, and analyzed this data in collaboration with my colleague Kelly Fraser at UCSD. All our genetic analysis was done in collaboration with Kelly. And so we observed 4 million variants in both these affected and unaffected siblings. And we used various filtering criteria to identify the variants that are likely to be associated with pathology. And we observed 8 variants in known retinal dystrophy genes, but they're not sufficient to cause disease. And we also observed 1 additional structural variant in the gene MERTK. So when we put together the information, all the variants, so we observed 2 variants in MERTK, which is a known IRD gene. One of these variants is a splice site variant, and the other one is a larger deletion that included exon 9 of MERTK. So these 2 variants, they could result in alteration of MERTK transcript. So all genetic studies which I'm going to present today, they were carried out by my grad student Pooja Biswas. So when we tested the segregation of these variants, so we observed the splice site variant is inherited from the mom, and the deletion variant is inherited from the dad, and her brother did not inherit both these variants. So these variants, so this suggests like it's likely to be, these variants are a possible cause of disease in this individual. So to further study the impact of these variants, so like initially, like if you both the splice variant and the deletion variant, so they could result in the formation of a short but functional multi-K protein, or it may result in complete loss of multi-K. So to study the impact of these mutations, so we used the iPSC model. So we have the blood samples of the father, mother, and this affected individual, and we generated iPSCs from the PBMCs, and we characterized them using standard methodologies, and we differentiated these to RPE. And these RPE were characterized for the presence of markers using marker like ZO1 and BEST1 and MITF. And we also established the polarized nature of these using BEST1 and ZO1 markers. So now that we have these cells, so we utilized these for further studies. And when we performed immunocytochemistry, so the red indicates multi-K. So you can see that. So there is a multi-K protein in the control line. This is the RPE differentiated from iPSC. But we did not detect the signal, multi-K signal in our patient line. So this could be due to two reasons. One is probably multi-K is not present, or the antibodies we were using did not detect the multi-K protein if a truncated protein is present. So we performed Western blot analysis using the iPSC lysates and also RPE. So one of the good things about multi-K is we can detect it in iPSC itself without going to RPE. But however, there may be different regulation, and especially in view of these mutations. So we tested its presence in both iPSC and also RPE. So we did not detect the presence of multi-K in the proband, but we detected the signal both in father, mother, and control individual. Similarly, we did not detect the multi-K protein in the patient and in RPE also. So to study the further, like whether there is a transcript, multi-K transcript present, so we measured the transcript levels, and the patient, so the levels of transcript are very low compared to her father, mother, and also the control sample. So this results in like possible loss of multi-K protein and transcript in this patient.
Radha,
Go ahead.
Can I ask a question?
Sure.
So when you plant the primers for detection of the transcript, where is it located, and whether did you try to detect the nascent transcript?
Yes, actually, just because of the in the interest of time, I did not include. So we performed analysis using multiple transcripts. So one of the ways we could also do is like upstream region transcript if there is a shorter transcript present. So when we use the upstream region primers, so then that will give us both the wild type and the mutant if the mutant is present and if the downstream sequence is altered. And then also, actually, maybe later I can show you, we could show the presence of transcript when we using the various methodologies. We could specifically extract but the mutant transcript, which is present at very low levels, especially the one with the deletion. So we could amplify and see the deletion exactly. But however, the levels is extremely low. And then of course, as you said, as we know, it's not stable. Yeah.
Thank you.
Actually, we do have a paper coming up, and then we have the details in that publication. I can also share with you. And then we tested the physiological impact of this variant on the cells by measuring transepithelial resistance. This was done in collaboration with my colleague Shyamanga Barwa. And so both patient and control lines, both of them had the TAR values were in the normal range. So we studied phagocytosis in these cells. So the control RPE generated from the control line and the patient line are shown here. When these were fed with fluorescently labeled photoreceptor outer segments, and then you can see the internalized photoreceptor outer segment signal in these in the control line, whereas we do not detect that, suggesting possible abnormality in phagocytosis in these. So, these studies show that like the mutations we detected possibly lead to loss of MERTK and impact the phagocytosis. And then, as we know, MERTK plays a critical role in RPE phagocytosis, and then that could underlie the disease mechanism. So this is one of the examples like where we established the functional relevance of mutations we identified. So this is another pedigree from Pakistan. As you see, there are multiple consanguineous marriages and multiple affected individuals. So this is actually a dream pedigree for geneticists because these are generally this makes our life easier to identify causative mutations because of multiple affected individuals and consanguinity. So we analyzed initially exome, which are shown with the red, and then we did not find any variants in the coding region, and then we performed the whole genome analysis, and once again we did not find any coding variants in the entire genome that could be likely possible candidates. So which was disappointing considering the size and the nature of this pedigree. So then we looked into the non-coding region, and we detected a novel variant in the upstream region of the gene TMEM216. So this is a known inherited retinal dystrophy gene. But the limitation here is, as you all know, we do not know the functional relevance of non-coding regions, and that is a major limitation, and particularly in case of eye tissue. So we are, hence we wanted to make sure this has something to do with pathology, or there are no other variants that are associated with the disease in this pedigree. So we performed homozygosity mapping analysis, and then when we carried out homozygosity, we observed about 14 megabase interval on chromosome 11 shared by all affected individuals and then not shared by unaffected individuals. So this is a region which is likely to harbor the causative mutation in this pedigree. So when we looked into this region carefully, so for genes that are associated with retinal degeneration or genes that could possibly cause retinal degeneration, are variants. So we did not observe any variants, any genes that are associated with retinal disease other than TMEM216, which we already identified before. And when we looked at the variants, again, that was the variant, only the 5' untranslated variant is the only variant we detected that could possibly associated with disease in this pedigree. So we performed segregation analysis, and then segregation shows this variant is present in all affected individuals and not in any of the unaffected individuals, suggesting its association with disease. So we looked into this a little bit more carefully, and then we are actually we have initiated studies to understand the impact of this non-coding variant. So this is an example, like we have several of these additional pedigrees like that with mutations in non-coding regions, and these require additional validation. So So here, this is another interesting pedigree we studied. So with one single affected individual, and when we performed whole genome analysis, we identified a mutation in the gene EYS, which is once again a known inherited retinal dystrophy gene. And this mutation is a nonsense mutation in the heterozygous state. So this will result in loss of loss of EYS. However, it's not sufficient to cause disease. So we looked for the second mutation in this gene, and then we have not found and we also did not find any other mutations. So at that point, we collaborated with Illumina to further analyze this data, and then we observed 2 5-base pair microdeletions. So one deletion is in the 5' untranslated region of this gene EYS, and the other one is in intron 12 of EYS. So once again, it's hard to interpret. We do not know what is the impact of these microdeletions in the untranslated regions and whether they have any impact or not, or associated with the disease or not. But initially, first we wanted to confirm the presence of these variants. So hence we designed primers from the planking, using the sequence, planking these deletions and sequence them. So it should be a trivial exercise, you amplify and sequence and you find these deletions. However, in this case, we failed to amplify this sequence using the primers using in the planking region. So at that point, we collaborated with Illumina and BioNanogenomics down the road from UCSB. So they have additional tools to examine this region. So Illumina could not come up with anything. We know there is something happening in that region, but however, we did not know, we could not identify. So BioNanogenomics has methodology to study large chromosomes and the whole chromosome as such. They use some technology which is somewhat a modified fiber fish, which we used to use in the past. So using this methodology, BioNano came back, and so they identified a large 1.6 megabase inversion, balanced inversion in this region. When there is this region is intact, but however, it is in the reverse orientation. So that is the reason we could not amplify when we used primers because it's in the wrong orientation. But when we sequence this and then map them to reference sequence, so the sequences map in the right orientation, like if you blast, whether they are in the right or wrong orientation, they blast to the right gene. So that's why we could not detect using the standard methodologies. So further, we have now we could establish the presence of this inversion. So this inversion includes exons 1 to 12 of EYS, and also it's part of the 5' region. But that will result in, this inversion will result in a loss of EYS protein on the transcript. And so we tested for the presence of these two in this pedigree. And so this affected individual has both the stop codon mutation and the microdeletion plus the inversion. Whereas the other two individuals have only the stop codon mutation. That's not sufficient to cause disease. So this is a challenging case for us. So we have additional example, something similar to it, this is the reason, although these cases have mutations in known retinal dystrophy genes, but they are challenging to find. And of course, make it more fun for us to work on this than standard finding some known or general mutations. So here, this is a pedigree where there are two affected individuals with inherited retinal dystrophy. And our analysis, whole genome sequence analysis, identified two mutations, one a nonsense mutation and a missense mutation in the gene IFT88, which is a flagellary transport protein 88, which is localized to the basal and proximal and distal ends of the connecting cilium in photoreceptors. And it's a very key protein in the ciliary transport and interacts with several other ciliary proteins. And mutations in IFT88 lead to retinal degeneration in a mouse model that has been shown. So, however, people have been looking for mutations in patients with retinal dystrophy in IFT88, and we have not identified. And this is the first pedigree with the mutations in IFT88 leading to retinal degeneration. So we wanted to test the impact of these variants simply because, there are. So what is the impact of this and how this results in retinal degeneration? So Pooja Biswas, my student, she collaborated with another postdoc in my lab, Anil Chekuri. Right now he's at Harvard. So Anil introduced these mutations in HeLa cells using CRISPR-Cas9 and isolated these different cell lines. And so the one that is expressing the nonsense mutation shows abnormal ciliogenesis and also loss of IFT88. Whereas cells that are showing the missense mutation, they show altered localization of IFT88, and some of them have cilia, but and then others also show abnormal ciliogenesis. So in control lines, we see the localization of IFT88 at the base and the tip of the connecting cilia. So the two mutations we observed in this patient, they result in abnormal ciliogenesis, and probably that is what is underlying the pathology. But however, Anil tested the transcript, measured the levels of transcript in cells that are with the stop codon mutation, and then so as expected, there is a significantly low levels of the transcript. And he also further showed that this transcript undergoes nonsense mediated decay, and so that's how it results in loss of this protein. So we established the involvement of IFT88 as a novel gene associated with inherited retinal dystrophy. So as I said, one of our major goals is to understand the genetic architecture of IRD. So Yucatan Peninsula, we studied 3 different pedigrees from 3 different populations. And in patients from Yucatan Peninsula have Native American ancestry. This is an admixture population, both with Native Indian ancestry and European ancestry. So interestingly, we identified mutations in 68% of cases, which is actually highest among all the 3. Although it is an understudied population. But about 50% of these variants we observed are novel, and 75% of them are not listed in any of the databases, and they were not reported before. And the NOMAD database is the database where variants detected in various populations worldwide are recorded. So it is possible these mutations came from the native Indian ancestry, and that's why this population is not well studied, and that could be the reason why we have not seen them before. And remaining mutations are from the European population. So that they are listed in the NOMAD database. So the other population we studied is from Pakistan. So this is the Asian population with it's an endogamous population with high consanguinity. The mutation detection rate is 60% in this population. However, 80% of the variants are novel here. So that shows the understudied nature of IRD in this population. And so we observed, but however, among these, only 14% are not listed in NOMAD compared to 75% here. So that is because, as we know, their South Asian population is reasonably well represented in the NOMAD database, where it listed all different populations. And so the mutations we observed here are specific to Asians, South Asians. And then the third population we studied is European population. Caucasian individuals from the US with European origin. And here we have the lowest discovery rate. So that could be because of the bias in the selection of pedigrees. The pedigrees that generally refer to us are pedigrees where others could not identify mutations, and then maybe it's challenging to find these mutations. And that could be the reason for low detection rate here. Among these, we observed 50% of them, again, high novel mutation detection rate. Once again, like that is probably the selection bias. And 50% of these are not listed in the NOMAD database. That is something surprising to us because European population and the Caucasian have been analyzed extensively. So it's likely that these are either private mutations or more recent mutations. So overall, our detection rate is about 60%. So in summary, actually, we observed about mutations in about 60% of cases, and 50% of the variants we observed are novel. So one of the interesting aspects in our study is 15% of the pedigrees we studied have atypical variants. Like as I said, I gave some of the examples. So large structural rearrangements, and then I did not talk about uniparental isodisomy. So in these pedigrees, the individuals inherited both chromosomes from one parent, not one from the mom and one from the dad. So we also observed de novo variants. So those are the ones, interesting to find and also very rare. And so that's why they were not identified before. So about 6% of pedigrees have mutations in more than one gene. And about 5% of patients and 5% of pedigrees have mutations in more than one gene there. In each patient, there are mutations, but one patient may have RPE65 mutations and also ABCA4 mutations. So these are very important to understand because in terms of treatment, so for this patient, it's not sufficient to have RPE65 therapy because they have other causative mutations sufficient to cause disease. So we observed a large number of novel variants, and after this analysis, at this point, about 40% of pedigrees that remained unresolved, and we suspect mutations in non-coding regions or challenging to find mutations in these. And at this point, that's the focus of our future work. So here I'm going to switch gears and talk about understanding the mechanism underlying retinal degenerations using late onset retinal degeneration as a model here. So I decided to talk about this because Chris and I share common interest in understanding this phenotype. So these studies were carried out in collaboration with Paul Sieving when he was in Michigan. So we started it way back in before 2000 in the late 1990s.
Sorry, I couldn't quite hear you. Could you please repeat what you said?
Somebody's Siri is answering. We have Paul analyzed a large number of patients clinically. And so these patients have dark adaptation abnormalities in their 40s and drusen and pseudoreticular drusen in their at early stage of disease. And they develop central vision loss in their 50s. And they also have, they develop neovascularization in their 60s. And their color vision is normal and ERG is normal. So they develop abnormally long anterior lens zonules in their 20s. And some of these individuals develop glaucoma in their 70s, but we are not sure at this point whether glaucoma is just a comorbidity or it is associated with a late onset retinal degeneration. So this is the pedigree. Initially, actually, before I got to Michigan, Dr. Sieving they have recruited these patients and analyzed them clinically and identified collected blood samples. And we collaborated and we analyzed the genetic basis of disease in this pedigree. Initially, we carried out linkage analysis and identified, eventually using various methodologies, so we identified a S163R mutation in the gene CTRP5 or C1q TNF5, segregating with the disease, and this gene was novel at that time. So this is a C1q tumor necrosis factor related protein 5, and I will be referring it as CTRP5, although the official name is C1q TNF5, which is a mouthful. This has a signal peptide and a collagen binding domain and globular C1q domain. The function of this protein is it's a secreted protein and it's present in the extracellular matrix, but however, the role of this protein in RPE is not known. So at that time, Mandal, a postdoc in my lab, so he carried out studies on this gene and the transcript, and he measured the levels of this transcript in various body tissue and detected the expression of CTRP5 in the ciliary body and RPE or posterior segment. So which is, we analyzed these two together, RPE plus choroid and in testis. We generated antibodies and localized CTRP5 to RPE and also ciliary epithelium. And we also detected the presence of CTRP5 in lens epithelium cells. So CTRP5 exists as a disystronic transcript with MFRP. MFRP is a membrane frizzled related protein. So this is mutations in MFRP lead to recessive RP and microphthalmia. So MFRP is a 17 exon gene, and CTRP5 is present in the 3' untranslated region of MFRP. And these two exist as disystronic on a disystronic transcript and then expressed in various tissue together. So we localized these two proteins in the RPE. So as you can see, red is CTRP5 and green is MFRP, and then they co-localize in RPE. Mandal performed, because these two are present in the same tissue, he wanted to see whether they interact with each other. And so he performed immunoprecipitation and then either with CTRP5 antibodies or MFRP antibodies. And we could pull each with the other, suggesting like these two proteins interact with each other. So to understand the function of this particular protein or impact of mutations in LOD, we generated a mouse model with the S163R mutation observed in patients. So we generated a knock-in mouse model. So here what I'm showing is these are the fundus autofluorescence images of this is the wild type mouse at age 20 months. And so as you can see here, in age-matched CTRP5 heterozygous knock-in mouse, there is significant increase in these autofluorescent spots. So we collaborated with Nahid Khan at University of Michigan on measuring the ERGs of these. And she measured the rod B wave recovery in these, like the normal ERG is normal in these mice, as we have seen in patients. However, rod B wave recovery is slower in these mice compared to the wild type mice, and then which, according to Nahid, is somewhat similar to the dark adaptation abnormalities observed in patients. So we collaborated with Monica Jablansky at University of Tennessee on to study the ultrastructure of these mouse RPE. And so here what I'm showing is this is the control mouse RPE at 21 months, and then it looks normal. And whereas in case of the heterozygous knock-in mice or the Lord mouse model, at 5 months we see abnormalities in Brooks membrane and also the presence of sub-RPE deposits. By 12 months, you see basal laminar deposits, or the basal infoldings are filled with electron dense material in these mice. Further, by 24 months, so this is the wild type mouse electron micrographs, and then this is the mutant mouse, that's a heterozygous mutant mouse model. And you can see that formation of deposits and more significant, the material that is filling this basal laminar or basal infoldings in these mice. And we also observed mitochondrial abnormalities. So when we performed immunohistochemistry, or we using the CTRP5 antibodies, so you can see at 18 months there is significant signal of CTRP5 compared to the age-matched wild-type control mouse. So this is so red is CTRP5 accumulating in this sub-RP region. So Shyamanga Barwa came to my lab a few years ago as a postdoc. He's a Fulbright scholar. So he came to spend a few years in my lab, and so he performed the studies which I'm going to talk about on this slide. So we obtained the retinal sections of LOD patients. Mainly we could get them through the collection of FFP, which this was collected by Dr. Ann Milam long ago. And so this is the age-matched control. So you can see the formation of deposits in this particular patient. And Shyamanga generated, we also have iPSC lines of these patients and unaffected siblings. So Shyamanga generated the RPE from these, and then so he demonstrated like the formation of these deposits in this iPSC differentiated RPE model also. And so since CTRP5 is present in the extracellular matrix and then it is accumulating the sub-RPE region, so we wanted to study if it interacts, the interactome of CTRP5. So we collaborated with Suresh Subramani and Kasia at UCSD on these studies, and we performed East-2 hybrid analysis. So this analysis, the top two candidates that came are MFRP, which we already know interacts with CTRP5, and the other one is HTRA1, which is a high temperature required factor A1 protein. This is a serine protease, and then known variants in HTRA1 shown to be associated with AMD. However, there is a contradictory information, and it is also, this is also right next to the ARMS2 locus. But however, this is a serine protease, and which is present in the extracellular matrix. Since we observed the East-2 hybrid studies showed interaction between HTRA1 and CTRP5, Anil performed immunoprecipitation he pulled down assays to see if HTRA1 and to test the interaction between CTRP5 and HTRA1. And then he has shown like, he could pull down HTRA1 with CTRP5 and CTRP5. So as you know, like Chris published an elegant paper in 2012 on crystal structure of the globular domain of CTRP5, C1q TNF5. And so using, based on his studies and additional information, so we generated we approached David Goodsell at Scripps Institute down the road from UCSD, and he generated model for the interaction between CTRP5 and MFR, HTRA1. So CTRP5 has a PDZ binding domain, and HTRA1 has a PDZ domain. And then, so the hypothesis here is, so they interact through this PDZ domain. So to test this, we generated constructs. So HTRA1 full-length construct and HTRA1 construct, just the PDZ domain and also part of the serine protease domain. So here what I'm showing is interaction between CTRP5 and HTRA1, just the PDZ domain region. And then you can see the interaction suggesting like that the interaction is through the PDZ domain, and having the PDZ domain itself is sufficient for this interaction. And both the wild type and mutant CTRP5 interact with the HTRA1. There is no difference between the wild type and mutant in that particular interaction. So we also studied like the role of the PDZ ligand motif in CTRP5. So we generated constructs with the full-length CTRP5 and then CTRP5 without the PDZ ligand. So when we tested, so we can see like wild type CTRP5 interacts with the HTRA1. However, when the PDZ ligand is deleted, motif is deleted in CTRP5, it does not interact with HTRA1, further suggesting the interaction is through the PDZ domain. So we know HTRA1 has a serine, it's a serine protease. So we measure whether we tested if CTRP5 has an impact on the activity of HTRA1. So we measured the activity of HTRA1 in presence of CTRP5. So for these studies, we collaborated with Carolyn Hayward and Chloe at Edinburgh, and so they provided the CTRP5 constructs. And HTRA1 constructs were provided by Peter Shah at UCI. So using the purified protein, we measured the activity. So in presence of CTRP5, so the HTRA1 shows enhanced activity. So both the wild type and mutant HTRA1, both of them increase the activity, enhance the activity of HTRA1, and then there is not much significant difference in the impact of these two on HTRA1. So now that we know CTRP5 enhances the activity of HTRA1, but HTRA1 is a serine protease. So we tested whether HTRA1 cleaves CTRP5. So when here what I'm showing is this is the wild type CTRP5 and the mutant CTRP5. This is the pattern you see without HTRA1. But when we added HTRA1, so HTRA1 there we see the presence of cleavage products when we added wild type CTRP5. So you can see the presence of cleavage products, whereas in case of the mutant CTRP5, we did not detect the cleavage products, suggesting like, so there is probably there is an impact of the mutation on cleavage of CTRP5 by HTRA1. So interestingly, around the same time, we were collaborating with Dr. Donna Garland at that time at Harvard on proteome analysis of sub-RPE deposits in these CTRP5 mice. So Donna already published similar studies on EFEMP1 mutant mouse model. So we initiated the collaboration with her, and then we sent the mouse eyes, and then she dissected and she performed proteome analysis. And so what she observed is she observed the accumulation of two different proteins in the eyes of these in the sub-RPE deposits. So she came up with CTRP5 and HTRA1. Those are the top two candidates she observed. And so she found that like this is the wild type mouse, CTRP5 at 5 months. And at this is the heterozygous mutant mouse. So there is significant increase, almost 10-fold increase here, if you see, in the levels of CTRP5. And in the homozygous mouse, so the levels are 18 compared to 0.82 you see here. Further, at 18 months, so the amount of the extent of accumulation is much significant both with the heterozygous and homozygous mutant mice compared to the age-matched wild-type control mice. So similarly, she also measured the levels of HTRA1, and they show the same trend. So the heterozygous mice show increased levels of HTRA1, and the homozygous mice also at 5 months show higher levels of HTRA1 compared to the wild-type controls. And with age, further, there is an increased accumulation. There is also otherwise increased accumulation just with age, but however, when you compare them with the age-matched controls, there is a accumulation of CTRP5 and HTRA1 in the heterozygous mice, which is the LOD model, and also homozygous mouse. So she looked for the specific peptides that are generated with the tryptic digestion, and then so this is the peptide that's generated around the mutation in the wild type, and so this was at 40% lower in heterozygous mice and absent in homozygous mutant mice as expected. So she also looked for the novel peptide that is generated because of the mutation, and this peptide obviously it was not detected in the wild type mice. However, it is and she detected this at high levels in the homozygous mutant mice in both cases, and then about 40 to 50% in the heterozygous mutant. So we validated this. We further carried out immunohistochemistry. We tested for the presence of CTRP5 and HTRA1 in the sub-RPE region of heterozygous mutant mice and homozygous mutant mice and compared to the wild-type mice. So in both cases, we see significant accumulation of CTRP5 and HTRA1 signal by immunohistochemistry. So since the CTRP5 and the HTRA1 are present in the extracellular matrix, so we tested additional extracellular matrix components that are associated with the RPE abnormalities. When we measured clusterin, ADAM9, vitronectin, and C3, so we see increased levels of these proteins in the extracellular matrix of the LOD mouse model. So as we know, like there are HTRA1 has several substrates. It's a serine protease, but the EFEMP1, which mutations in EFEMP1 lead to Doyne's honeycomb dystrophy, which is a deposit-forming phenotype, and similarly, mutations in TIM3 lead to Zorb's fundus dystrophy, and C3 and vitronectin, which both are associated with AMD, and all of them show this deposit-forming phenotype, and similar to LOD, which is due to CTRP5. And all these are substrates of HTRA1, suggesting a common thread in the pathology of these diseases and a possible role for HTRA1 in this disease pathology. So our study showed like CTRP5 modulates the activity of HTRA1 and HTRA1 modulates ECM composition, and alteration of HTRA1 activity impacts ECM composition, at least in CTRP5 mice. And LOD pathology probably involves HTRA1, and HTRA1 may play a role in the deposit forming phenotype. So obviously the studies we carried out were not done just by my laboratory. So we collaborated with a large number of collaborators, actually worldwide. So all genetic studies were carried out by Pooja Biswas, a graduate student in my lab. Most of the studies are presented today. And Anil Chekuri carried out the functional studies of LOD mouse model and LOD studies, and also IFT88. And Shamanga Barwa, who was my former postdoc and currently he's my colleague, assistant professor at UCSD. So we continue to collaborate. And I mentioned our interaction, our collaboration with other investigators on UCSD campus. And so Murthy Chavali characterized the CTRP5 mouse model, and he's currently an assistant professor at UPenn. And Navadesh Mandal, he carried out early studies on CTRP5. He was when he was a postdoc in my lab, and currently he's an associate professor at University of Tennessee. And I also collaborated with a large number of clinical collaborators, and some of them who contributed significant number of pedigrees are Jackie Duncan from UCSF and Paul Saving now at UC Davis, and John Heckenlively and Carrie Branham from University of Michigan, Ada Villanueva from Mexico, mainly she contributed all the pedigrees from the Yucatan Peninsula and Mexican pedigrees. Amir Riazuddin is my long-term collaborator from currently from Johns Hopkins. Amir and I did postdoc with Fielding's lab at NEI, NIH, and then we collaborate on various genetic studies and mainly on analysis of Pakistani pedigrees. Astra, we have been collaborating with Astra on gene therapy studies. So she generated the constructs for both for MFRP and CTRP5 gene therapy studies, and which I did not present today. So these are some of the members of my lab, and not all of them. And the next steps for our studies are, as I said, like we will focus our studies. We are focusing our studies on the mainly the elusive genes project, which is focused on challenging to find mutations using various methodologies and different models, and understanding LOD and deposit forming phenotypes. Once again, mainly because this is the age-related disease, so we are studying this aging phenomenon within the context of aging, using CTRP5, MFRP, and HTRA1 mouse models we have, and also iPSC models. So I will be happy to take any questions.
Thank you very much. Bravo. So let's go to questions. Ivan, do you have a question here? Hello, Ivan.
Hopefully.
Maybe he will connect soon. How about Philip?
Very nice presentation. So I had a question that I typed in the chat on the first part of your talk related to the MERTK mutations that you identified, which are really interesting. I was just wondering, is there enough known about the structure of MERTK? I was just wondering how much you guys kind of knew in advance about that, as far as what the feasibility was of being able to remove an entire exon and have a functional protein produced from that.
MERTK is a very well-studied gene as a gene for inherited retinal dystrophies. But however, we do not really know the impact of the deletion of that. And actually, we do. I do not have it handy, but we have different models. We predicted based on the deletion, that particular deletion, whether we can, what kind of truncated protein it could generate. So there is always a possibility, and in this particular case, luckily for us, we have the cell iPSC lines to test some of those hypotheses.
Can I get one more in really quick related to the HTRA1? So I think I missed this point. Do you think that in the case of the mutation that this S163R, I think, mutation that you talked about in the second part of your talk, do you think that inhibition of HTRA1 like activity there would be protective against the phenotypes that you see with that mutation, or have I got it mixed up?
I could not figure out, to tell you the truth is, we do not know. I think we try to see which would be the appropriate hypothesis, like whether increase or decrease is the one which is which may have an impact. So I do not have a good answer to that question at this point based on the data we have, and hopefully we will be able to generate. And we also have actually additional mutations in recently observed in patients with CTRP5, and then we are generating the iPSC and RPE. So that first actually will tell us whether HTRA1 indeed has any impact on CTRP5 associated disease, or one of my major concerns is it's a protease and it's present in several cells. So we need to understand further to be able to address these questions.
Thank you.
Ivan, are you still there from our institute?
Hello, I'm here. Yeah.
Do you have a question?
I have a question. Thank you for the wonderful presentation. Really, really beautiful.
Thank you.
I have a question about your IPS protocol generation. Which human blood cell do you use for generation of your IPS cell to further differentiation to RPE cells?
PBMCs.
Oh, PBMCs. Oh, okay. Yes. And about the no-coding region in your PKRD307 variants, are they transcribed, meaning are they no-coding RNAs or just DNA distal enhancers controlling promoters?
Can you repeat that question, please? The second
it's a question about PKRD307 variants, and you mentioned that it's not a transcribed gene, and I was wondering if it's a no-coding RNA or just a DNA distal enhancer region controlling promoters. I know you are not doing, you're focusing on the coding genes, but you also mentioned no-coding regions of the genome.
Yes, actually the variant is first TMEM216 is the gene downstream to this variant we observed, and the TMEM is a coding gene, and it's
coding.
Yes, it is involved in causing ciliopathies. So mutations in that gene lead to retinal degeneration. It's a known retinal degeneration. So
okay,
and then, yes, you are right. We have a study ongoing at this point on understanding the impact of these non-coding sequences using single-cell genomics in a human retina. So.
Yeah, I was glad to hear that because it's a very fascinating and unknown territory there, and it seems to be more and more important and relevant for human diseases and mutations.
Exactly.
Thank you.
All right, thank you. Let's just move on to Arun and Alex. Arun.
Hello. It was really a nice talk, and then regarding your second part of the talk, I have a question. So since here you show that CTRP5 is, related to causing the late onset RD through HTRA interaction. So, what will be your future therapeutic approaches? For example, is it possible to rescue CTRP5 knocking mouse model phenotype using maybe like HTRA1 inhibition?
So actually, thank you for the question. That's an interesting question. So at this point, we are trying to establish the role of HTRA1 in LOD pathology using, as I mentioned, other mutations. So that's first thing we are doing at this point. And we also have HTRA1 mouse model and CTRP5 mouse model, and then so we are also testing whether we can start. We are trying to address the question you asked whether HTRA1 has what is the impact of HTRA1 presence or absence in CTRP5 So we are hoping they may help us in addressing the questions you raised, because the eventual goal is to think about what is the best way to treat these diseases, and we hope the knowledge we gain through our studies contribute in that direction. But at this point, the information we have is not really clear to say we can knock off HTRA1 and then protect CTRP5 or LOD disease or drusen deposit forming disease. We are not there yet.
Okay, thank you.
Hi, this is Alex from Dr. Palczewski Lab. I have actually two questions. One, since CTRP5 interacts with MFRP, did you check whether HTRA1 actually cleaves MFRP or has some influence on MFRP? And
go ahead.
Yeah, no.
So MFRP is a membrane frizzle-related protein. It's a membrane protein. So hence we have not tested it. CTRP5 and HTRA1 are present in the extracellular matrix. So that's why we have tested. But we do have, I have not presented studies like we are carrying out studies on the interaction of CTRP5 and MFRP and then role of MFRP in LOD pathology. So we have those two mouse models, and then so that's another study which I did not talk about, but we are trying to learn about it and then see whether all these three have a how we can integrate the information we are generating through these different studies to understand. Mainly, the goal is to understand LOD pathology.
Yeah, so my understanding is that actually most of the MFRP protein is exposed to the extracellular space, so that's why I was bringing up the question.
Exactly.
And the other one, more general, do you have any thoughts on the biological relevance of this interaction between CTRP5 and MFRP? Since, my understanding is that the phenotypes that we observe in patients who have mutations in both proteins are to some extent actually different. Yeah, they are not that much overlapping.
Yes. So I think also MFRP disease is a recessive disease. So and another interesting aspect is CTRP5 is a similar to adiponectin. So that brings a different dimension to the whole story here. And CTRP5 has also been reported to be it is an activator of AMP kinase. So we are carrying out some studies. So CTRP5 interacts with MFRP, and then also it binds MFRP, and then adiponectin also binds MFRP. So there are studies in the context of that. So we are carrying out studies to understand. So once again, we do not have a clarity on the role. Maybe CTRP5 acts. CTRP5 is also present inside the cell. It's not only outside the cell. So and the intracellular CTRP5 has been shown to have AMP kinase activator of AMP kinase. So that's once again, these studies are the role of this CTRP5 is not well understood, and which one is the actual which activity is actually leading to pathology or contributing to pathology in LOD is not at this point.
Thank you.
This was a really wonderful presentation and wonderful data. And with the final question and comments, we're going to Dorota.
Thank you, Chris and Radha. It was amazing talk how you went from the genomics to molecular mechanisms. Thank you. So actually, I would like to come back to the HTRA1 protein for a second. So there is this recent paper in Immunity talking about the monocytes bringing a lot of HTRA1 to the space. So how do you think the role of this? I kind of see it as an abundance of HTRA1 that can interrupt the internal interactions between these proteins. But what is your view on this problem or data?
So, based on what we have seen, one of the hypotheses is, so we have high amount of HTRA1 that's accumulating. We also see high levels of CTRP5 accumulating, and then both wild type and mutant are accumulating, and both wild type and mutant can activate HTRA1. So now we have high levels of all these, and then so probably we have increased activity of HTRA1, which is modulating the composition of extracellular matrix, and that may be contributing to disease. So that is one of the hypotheses.
Yeah. Thank you so much. It was fantastic talk.
Thank you very much, and I will see you all and many others next Friday.
Thank you.
Thank you.
Thank you, Chris and Dorota, for the invitation, and I enjoyed sharing our data.
Radha, I always wanted, you always give such a great talks, and I knew you will tell us something new, so it's really great. And